Today we're going to cover the autonomic nervous system. So I just want you first of all just to have a look at the structure. Okay, that's the first thing to do. So here you can see your spinal cord. Okay, so you can see the H shape. That's the or the butterfly shape as it's sometimes called. That's grey matter. That's processing. That's part of your central nervous system, obviously. And the white matter. That's the communication. Okay, so computer is the grey matter. White are the cables. And then, of course, you've got the various routes coming out, the various nerves coming in and coming and going out of the spinal cord. So other things to really note in this diagram is that you can see that the sympathetic trunk flanks alongside the vertebral column, okay, because you can see that ventral column and you can see the ribs in the diagram. And so remembering how information is processed by the central nervous system and that spinal cord. Sensory information goes in through the dorsal route, motor information or efferent information comes out through the ventral route, the front. So if we're following the pathway that's coming out, all this information, whatever information has been processed, and your preganglionic, we're thinking about the sympathetic nervous system at the moment, okay, so we're only on the sympathetic nervous system. The preganglionic fibers coming out arise from the lateral horns in T1, remember, to L2 via the ventral route. So here they are, they're coming out. And here they are coming out. And then the axons go down the white rami communicans. So just follow my arrow for now, even though the red pictures come up elsewhere. Follow down, comes along here, and down the white rami communicans. Same thing, okay? It's come out at this level, hidden, you can't see it so much, coming out and down the white rami communicans. It therefore is now entered into the adjoining sympathetic trunk or the sympathetic ganglia. So here are the big bulges. They're your sympathetic ganglia. Okay, and you can see those because they're bulges and they've got lots of cell bodies in there. So they've entered the sympathetic trunk, this whole thing. And this preganglionic fiber then synapses either in this sympathetic trunk ganglion in each one of these. So it comes down, synapses, comes down, synapses. Comes down, synapses, okay, potentially. Or it can pass through the trunk ganglion and synapse in a distal collateral ganglion, as they're called. And we will cover that a little bit later. But for all intents and purposes, it's coming out through the ventral route, down the white rami communicans, and into the sympathetic trunk, where it can potentially go on to synapse or synapse very close by. So when we're thinking about that, we've got to think about how and where it's going. So we've got to think, first of all, what are the pathways to the head? So this is where your sympathetic preganglionic fibers emerge. So they're going to the head, remember. They emerge from T1 to T4, so we're at the top region, thoracic 1 to 4, and ascends, so watching it coming up, ascends the sympathetic synapse to synapse with postganglionic neurons. So there we are. So they've come up and they've gone up as a general rule. They've come in, they've come up, and there we're going to the head. T1 to T4, up to there. So they've ascended the sympathetic trunk, synapse with the postganglionic neurons, which then deliver information. Okay, tell the eyes what to do, or the lacrimal glands what to do, or the nasal mucosa what to do, whatever it might be. Okay. So what does it actually do in each case? Well, of course, it stimulates the dilator muscles of the iris. So, in other words, your pupils dilate. Okay, and if your pupils dilate, you get more processing power. It innervates the smooth muscle of the upper eyelid. In other words, it's lifting up the upper eyelid. It inhibits the salivary glands, hence why your mouth goes dry when you're nervous. And it innervates the skin and the blood vessels of the head. Okay, so it innervates the skin and blood vessels, allows them to dilate appropriately or constrict appropriately. Thinking about when you get nervous and cold and clammy, you vasoconstrict, but later on as you start to get too hot and really, really hot, then it'll start to dilate. So it's to try and lose heat. So there are different processes going on, but initially you'll vasoconstrict to keep more blood to the systems that need it. In other words, your skeletal muscle and your heart. And of course, it inhibits the other glands, so the lacrimal and the nasal glands. Now, if we're looking at the pathways to the thorax. So in this case, your sympathetic preganglionic fibers, they're emerging from T1 down to T6, and they go on to synapse with your postganglionic neurons, and they come in and move in through very complex plexuses, your cardiac and pulmonary plexuses, and they can synapse then with the heart and the lungs. And again, we're showing the preganglionic neurons and the postganglionic neurons. And of course, what does the sympathetic nervous system do to the heart? Increases heart rate, increases contractility, so increases stroke volume. And in the lungs, obviously, dilation of the bronchioles and increased breathing rate through various mechanisms. So what about those collateral ganglia that I just mentioned a couple of minutes ago? Well, the sympathetic preganglionic fibers that emerge lower down, so from T5 down to L2, they synapse with postganglionic neurons in what are called collateral ganglia. So these are postganglionic, and then the postganglionic neuron goes on to synapse with the effector organ. So again, what I want you to do is just have a little look at what is actually going on in this picture, just to try and understand it. So have a look at the picture, look really closely for the actual synapses. Okay, you need to know where those synapses are and therefore where the ganglia are. So again, what you should have spotted from this, these lower regions, T5 to L2, you can see that they come through. Okay, they go through. In this case, if we're looking at these ones, you can see that they go straight through the sympathetic trunk. They don't synapse in there. They pass straight through, and then they come in and they synapse with a distal ganglia. It's called a collateral ganglia. Okay, and they all have different names. You don't need to worry about it for now. And so when they synapse in there, you then have your postganglionic neurons going over to the liver, gallbladder, the stomach, the spleen, the kidney, and the rest of the small intestine. And so the important point is you've got these collateral ganglia that are present. And as a general rule, they primarily inhibit the muscles and the glands in the abdominopelvic visceral area. So in this region, apart from the adrenal gland, of course, which is there, they inhibit these structures, okay? Because you don't need your GI tract or your kidneys to work particularly well, or your gonads, your genitalia, to be fully functioning during a fight or flight response. Okay, remembering, of course, what's going to the adrenal medulla, you can follow it backwards if you want to. From this case, you can follow it all the way backwards, and you can see that it doesn't synapse anywhere. So it's only got a preganglionic fiber, stimulates the release of adrenaline and noradrenaline. Okay, so a couple of questions for you. So I want you to look through these questions, and in each case, is it true or is it false? So again, please press pause and start it again when you think you've got the answer. The white rami communicans contain which of the following fibers? Preganglionic parasympathetic fibers, postganglionic parasympathetic fibers, preganglionic sympathetic fibers, or postganglionic sympathetic fibers? I'll give you a clue, only one of them is correct. Yeah, it's the number 3, the preganglionic sympathetic fibers that have left through the ventral root, come down the white rami communicans and into the sympathetic trunk. So the collateral ganglia are involved with innervating which of the following? So I just want true or false in each case, and we'll run through them. So abdominal organs, correct. It's the thoracic organs, incorrect. Head, incorrect. Erector pili, incorrect. It's the sympathetic trunk ganglia that innervate the last 3, the thoracic organs, the head, and the erector pili. So what we're going to just finish on, we're just going to look at some autonomic or visceral reflexes. So visceral reflex arcs have the same components as the somatic reflex arcs. So when you're thinking about the knee-jerk reflex, for example, or moving away from moving your hand away from a hot object or a sharp object. But a visceral reflex arc has got a 2-neuron motor pathway. So when you're thinking about these autonomic or visceral reflexes, you've got to think about what are they responding to? Well, there are many examples. So your visceral sensory neurons may detect chemical changes. They may detect stretch, irritation of the viscera, so the internal organs. And examples include things like emptying the bladder and emptying the rectum. But really, when you're thinking about what's going on, I want you to really be thinking about the stimulus. A stimulus comes in. You therefore get a sensory receptor in the viscera. This then comes in through the dorsal root, so through the back, dorsal root ganglion, synapses in the spinal cord, and then you've got your viscera, so that's your visceral sensory neuron, comes into your integration sensor, okay? Maybe a preganglionic neuron, which is what is actually shown, maybe a dorsal horn interneuron, because you often get interneurons between the two, and it may be within the walls of the GI tract, because the GI tract has its own specific enteric nervous system, so it's got its own nervous system, but it does engage with the central nervous system as well. And so then you get the efferent pathway coming out. And so what you can see is the efferent pathway, which is a 2-chain neuron. We've just been talking about it. It's coming out through the ventral root, okay, comes down through into the autonomic ganglion, where it goes on to synapse, and it then goes on to the postganglionic neuron, to the visceral effector, where you get a response. And so we've given you one example. There are lots of examples. Your light reflex, your thermoregulation. So when you get too hot or you get too cold, you have these reflexes. Okay, and the light reflex is another one. Remembering, you don't have any control over these reflexes. So emptying the bladder or emptying the rectum, you do get smooth muscle dilation. Okay, but there is also some somatic innervation there, which is why you can control it. But you are getting the autonomic reflexes going on as well. So if we look at one little example, or sorry, another one just to show the concept of it rather. So here we have the afferent neuron. So we're getting sensory information coming in with receptive endings, passes through the afferent peripheral nervous system to the CNS, where you generally have interneurons, then onto the autonomic preganglionic neuron, synapses in a ganglia, and then your postganglionic neuron hits the effector. Nice and simple. And so a great example is this sympathetic reflex in thermoregulation. Remember, you have autonomic reflexes, can be sympathetic or parasympathetic, of course. You may get an increase in your body temperature, so you're hot. You get increased activity within your thermoreceptors, okay? So therefore, this reflex increases the sympathetic discharge to sweat glands. So in other words, you've got an increased body temperature, so it stimulates increased sweating. The reflex decreases sympathetic discharge to your cutaneous blood vessels. That's the blood vessels just underneath your skin. So you're getting decreased sympathetic activity going to those blood vessels. And so what that means is you get decreased vasoconstriction. And if you've got decreased vasoconstriction, it kind of suggests, but we'll say it for now, that it's causing vasodilation. It's not actually causing vasodilation, you're just getting less vasoconstriction. But the whole point is you're getting a vasodilatory effect, which means more blood is going to the skin surface, which means you are cooling yourself better. So that's a great example of a sympathetic reflex in thermoregulation because you're too hot. You sweat more and you look redder, okay, which is allowing heat to be lost. We then have a parasympathetic reflex for you to think about. And this is the pupillary light reflex. Okay, and again, it's a homeostatic autonomic reflex. So again, what I want you to do in this one, I just want you to have a little look first. Okay, have a look at this diagram and tell me what you think is happening. We'll start with light coming in, and we're thinking it's a parasympathetic reflex. So think about what's going to happen to the size of the pupil is the first thing for you to think about. And think about where the light goes to. The light goes on or stimulates neurons within the retina. Okay, so I want you to have a quick think. What's going to happen? What's going to happen to the pupils? So light comes in. It stimulates a variety of pathways in the retina, and we haven't done the special senses, and eyes are one of the 5 special senses. We'll deal with that at a later date. But it stimulates nerve cells within the retina. That eventually gets processed and passes through your optic nerves and moves into the Edinger-Westphal nucleus, so this region here. Okay, and some of that information goes to the brain and to the visual cortex, where you can start to process that information on a conscious level. But you also have the interneuron present and then you have your preganglionic neuron. Preganglionic neuron is coming out, it's parasympathetic, cranial nerve 3, comes into the autonomic ganglia, into our postganglionic neuron, and it synapses on both eyes. That's the important thing I want you to be thinking about here. So if you do a pupillary reflex, if you shine light, if you block light from one eye and just shine light into the other one, you'll stimulate that one eye's retina, because you're not stimulating the other eye's retina, stimulates this whole pathway, comes along here, both pupils should constrict. Okay, it's a good little clinical test for you so that you know that the Edinger-Westphal nucleus is working correctly and all the various eye processes are working properly. Shine light in one eye, both pupils should constrict. And there's a great example of a parasympathetic homeostatic autonomic reflex. So last thing I want you to do is fill in this table. Okay, you should be able to do this now. I want you to, this is kind of a summary of some key features or key structural elements, if you like. So, origin. Where does the parasympathetic nervous system originate? Originates in brainstem 3, 7, 9, and 10. Remember, 10 is vagus, and your sacral S2 to 4. Remember, you should have paused this so you can think about it in each case. Sympathetic nervous system, T1 to L2. Where is the ganglia located? Parasympathetic, it's either within the visceral organ or very close to the organ that it's serving. Sympathetic, it's either close to the CNS in the sympathetic trunk or within a collateral. And we talked about those, how the collaterals are delivering the information through to generally the abdominal organs. Preganglionic fiber length in the PNS is long, in the sympathetic is short. Postganglionic fiber length, short parasympathetic, long sympathetic. Finally, the functions, crudely put, maintenance for the parasympathetic, prepares the body for activity in the sympathetic. And these are things that I've asked you to think about right now, but also that we will cover later on.